Okay, we're live on YouTube, and good. And then we should be live now on LinkedIn Live. I'd like to welcome everybody to this MedVR talk, part of a series of talks we've been running for the last 2 years. I think this is talk number 41 that we've held. We run a conference in small bites over the course of a year. MedVR is focused on innovation in medical XR and research and development, both applied research as well as foundational research to identify what big breakthroughs need to be made to advance the field. We are working on an accelerator that will open up in the Longwood medical area of Boston to work directly with physicians and scientists and engineers and universities, which is our primary goal. And of course, keeping up with these talks is important. While the field of our talks might change from week to week, we might go from psychiatry to surgery. What they have in common is they tell the story of the journey of a medical XR innovator, which where we can learn quite a bit from. I am really excited by this talk. When I saw the paper behind it, it really pleased me because it's an application specific headset, and I keep wondering why we don't do more when in terms of headsets. When you look at what fields are maturing in medical XR, it's training, pain management, and psychiatric innovation. And it's really only because these applications fit in today's headsets, and getting the conversation over to tomorrow's headsets is really important. And this is one of those talks. Our moderator today is Jeff Bale, who is the manager of the user interface at Vicarious Surgical. He's had over 2 decades of experience in simulation and augmented and virtual reality that he's worked in, as well as being a leader here in Boston in the community, bringing the community together to advance our goals. So with that, I'm going to turn things over to you, Jeff. And but before I do that, one housekeeping point, folks, is there's a Q&A chat for submitting questions. There's also a plain old regular chat. Please use the Q&A chat to submit your questions. I'll be shepherding the regular chat and remind people to go over the Q&A chat. But Jeff, it's up to you.
All right, thanks very much for the introduction. And before I go ahead and introduce our speaker, I'd like to start by inviting all of you to join the FDA, industry leaders, and the Medical Device Innovation Consortium for a 2-day regulatory science conference It's called MXR 2023, and it's taking place October 24th through the 25th in Bethesda, Maryland. We're going to bring together leaders in XR who are focusing on healthcare applications, learn about technology and advances in XR technology, current medical applications, drivers of innovation and adoption, and most importantly, recent regulatory advances in the field of medical extended reality. So this is really an extension of a workshop that happened coming up on 4 years ago now at FDA headquarters. There was a one-day workshop in medical XR. Actually, Stephen and I were both there, and we were handing out invites for what was to be the very first MedVR hackathon. And then less than a week later, COVID hit and we were in lockdown. And unfortunately, that put the stop to the hackathon. However, the important work that started that day at FDA was carried forward by MDIC. And all the different subgroups will be there to present, as well as industry leaders and some great speakers. So I really hope you can come join us for this event. I think it's going to be really important to, kind of reveal the future of the potential and the regulatory landscape for this technology in medicine. So you can learn more at mdic.org. I'll also drop a link to the event directly in the chat. But now I'd like to introduce our speaker, Leo Shmulevich, PhD, is an assistant professor of dermatology, medicine, radiology, and pediatrics, and a principal investigator in the Biophotonics Research Center for the Mallinckrodt Institute of Radiology at Washington University School of Medicine in St. Louis. Shmulevich is a physician and scientist with a weekly pediatric dermatology clinic, and he leads a multidisciplinary vascular anomalies clinic. His research is focused on developing novel imaging technologies to help address unmet needs facing patients' skin disease, including skin cancer and inflammatory conditions. I know someone asked for a link to his paper. I'll do my best to find that and drop that into the chat. But also just wanted to reiterate what Steve said. If you have questions, I'm going to be looking for them in the Q&A section of Zoom. So please drop them in there and I'll do my best to address all your questions at the end. And with that, let's hand it over to Leo.
Thanks so much. Hopefully you guys can see my screen. It's being shared. Is it working?
Yep.
Okay. All right. Thanks so much for the introduction. Thank you for the opportunity to speak. The paper is a preprint. It's still under review. I'll show some figures from that preprint during this talk. And you can find me on Twitter as well. I'm happy to connect by email, by Twitter, however you like. So the work that I'm talking about today is work I did really during my postdoc and work that I hope to continue to pursue as a professor now. I just want to start because oftentimes people, things get a little bit hectic at the end of a talk. And so I would like to start with acknowledgements. And none of this would be possible without my mentor, Dr. Samachilafu, who's really the visionary behind so much of this work. He was the director of the optical radiology lab at WashU here at the time of the work. He was then brought over to UT Southwestern to become the inaugural chair of biomedical engineering. And that's where he is now. And that's where a lot of the work for this continues. And then the other person is not Dr. Samachilafu, but Dr. Christine O'Brien. There's a typo here. And she was a postdoc with me at the time, and we did a lot of work together, and she's a professor here at WashU in biomedical engineering now. And so we did a lot of this work together as a team. So what I show you is really the result of a lot of people putting in effort to make real the vision that our mentor, Dr. Chilafu, has. So I want to start around just centering around what it is that we do. And the focus of what we do is called fluorescence-guided surgery. And people who are part of this session today probably know this concept pretty well. But the idea is, if we could somehow find a way to visualize cancer or visualize disease in a way that our eyes do not see, so to enhance and augment our vision, then we could improve surgical outcomes or improve outcomes in terms of treatment. Sometimes a surgeon goes in, they try to find a tumor, they can feel it because the tissue feels different, but they can't see it. They don't exactly know where the margins are. They go and do a surgery based on guidelines, based on a guess, based on a rough estimate, and that doesn't necessarily ensure that all the tumor is removed. Or sometimes they may go into a surgical field, and there may be a lot of normal anatomy around nerves and vessels. And sometimes the cancer makes it very difficult to operate, and you end up cutting a nerve by accident. So there's this kind of general sense that, gosh, if we could enhance our vision, if we could make a way for the nerves to glow or for the tumor to glow, that would really make surgery and more effective when it comes to surgical removal of tumors, which, in many ways is still the best way to treat a cancer. So the concept is, let's go ahead and inject some compound that will target the tumor through some biology, through some chemistry. And then when you stimulate that compound with light of a certain wavelength, then that compound will have a fluorophore within it, and that fluorophore will be excited and will release some light, and then you can detect it and you can display it in some way. And one of the neat ideas in this field is to use so-called near-infrared fluorescence agents, or NIR, as I have here. And the reason for that is that near-infrared, it's invisible to our eyes, but it can be visible by cameras. And because fluorescence is fairly weak, if you try to do fluorescence with something that fluoresces in the visible, then your eyes won't pick it up when the lights are on, because the lights kind of dominate the signal. So the advantage of doing near infrared is that you can have room lights on while also doing near infrared imaging. And this idea has been around for decades. There are multiple commercial devices available, and this very nice review in JBO has, lists many of these devices. They're overall fairly bulky. They're quite expensive. They're the kinds of things that you have at a center like WashU, a place that has a lot of resources and that is a high-end medical center. And it kind of came up in my mentor's mind that this technology is so powerful, but it's not accessible to the world. It's only accessible to the lucky few. So the concept that he came up with, and also in addition to that, a lot of these devices, the way they work is there is a device that shines light, there's a camera, and then there's a screen on which you see the fluorescence or you see the signal. That's not the best interface potentially for a surgeon who's working in a low resource area, or just a surgeon in general, because the surgeon is working in a surgical field. They're looking at what they're trying to do. They don't want to be constantly looking up at a screen, looking down, looking up at a screen. It's not so efficient. So his concept was, can we do this in a virtual or augmented reality approach by having the same kind of idea? You have a tumor, you target the tumor, you excite the light, you detect the light, but then you process that and stream it into some sort of heads-up display. And the initial version of this was a virtual reality display. So you would see a superposition of both visible and near infrared superimposed on each other. In many ways, this is kind of like what, as far as I understand, Vision Pro is, the new device, which is kind of an augmented reality approach in the sense that you see the real world, but you're actually not seeing the real world. You're seeing a reconstruction of the real world, but then you're also adding in this invisible thing, which is the near infrared. And the initial results on this was really exciting. They had a few patients that they tested it on, 10 patients with breast cancer, 5 with melanoma. They were all undergoing sentinel lymph node biopsy. And they had gold standard imaging done, which is you inject methylene blue with a radio tracer and you visibly see if the thing is glowing blue, you say, aha, that's a positive tumor. Well, this device was able to detect fluorescence in the sentinel lymph nodes in all cases that were positive, but this is showing one case where it wasn't really obvious to the surgeon that this thing was glowing blue, but our device was able to pick up the fluorescence, and indeed this was a positive sentinel lymph node. So this was an exciting result from almost 10 years ago with this virtual reality-based system. And then there was an upgrade to the system of a see-through display, where again, you had kind of, these see-through glasses that also were able to project an image onto your view. And there were multiple cameras to try to capture that image. And one of the cameras was visible and one of the cameras was near infrared. And again, this was a study of about 3 breast cancer patients. They were undergoing lumpectomies or mastectomies, and you could see the tumor glowing with a fluorescent targeted agent. And you could also see the tumor cavity glowing with a fluorescent targeted agent. And in one of 3 patients, they actually saw this glow in the tumor cavity, and then it turned out that that was the one patient that had positive margins. So the device was really able to detect something that by eye you could not appreciate. The surgeon would not necessarily know that the cavity still had tumor in it, but the device was able to find it. So this was really exciting, but this was done, 7 years ago, and our lab is working on it, trying to improve it. But every time we tried to make changes, make adjustments, we still were left with what was fairly bulky, what was kind of hard to iterate, hard to rapidly prototype. And Sam one time came to my lab, because my lab is full of like 3D printers and all sorts of stuff, because I love making stuff. He's like, Leo, can we rethink the system? Can we kind of redesign the system in a different way? And, I said, sure, let's apply kind of the maker movement to it. Can we take what is so exciting now to me in the world, which is that, suddenly we've democratized a lot of things that before were very hard to do. There's cameras, there's computers, they're all wearable, they're small. Are they powerful enough to create a device like this? And importantly, can we do that to make really the vision that he had from the beginning, to take these devices that are hundreds of thousands of dollars now and make them accessible to the world? So this was the question that we had. And we started with a very popular tool of the maker movement, which is the Raspberry Pi system. The Raspberry Pi is a tiny little computer that is the size of a credit card. It's $50. It can be battery powered. And it comes integrated with these camera modules that you can use. People use them to build, DIY bird feeders that trigger whenever a bird comes up and you can watch the bird fly. And actually at the airport, I once noticed that the departures at our St. Louis airport gate, the actual screen had a loading screen from Raspberry Pi. So I guess, the airports are using Raspberry Pis now. Everyone's using Raspberry Pi. So the question is, can we use it for our purposes? The first kind of glimmer of hope that we had was just a question of, can we detect indocyanine green, which is a FDA-approved near-infrared fluorophore? Can we detect it at low concentrations using the Raspberry Pi system? So we looked at this Aptina CMOS sensor. This was the device that we were using in those various trials with the breast cancer patients and the melanoma patients. It really could detect down to about 100 nanomolar of this ICG compound. And this was a custom built sensor that, people spent years trying to make back in 2013. We compared it to a $500 scientific high, like 16-bit or 12-bit camera. I think this was it comes in two different features. And it, using this monochrome camera, it gets very hot. It's like only for scientific purposes, but this thing could detect 1 nanomolar pretty reliably. It was pretty impressive. So then the question is, what about a Raspberry Pi camera? Here's this Raspberry Pi camera with an extension cord to allow the sensor to be disconnected from the board itself. What can this thing do? And we were really surprised, and there were, one of the members of our lab lost a bet because she bet me that there's no way that the quantum efficiency of the sensor would allow anything below 100 nanomolar, but we could do 100 nanomolar, and we could see a signal at 10 nanomolar, and we could even see, it's noisy, but we could see a signal above background at 1 nanomolar. And that was really surprising to us, that this $30, cell phone hobby type camera, even though it doesn't have great quantum efficiency in the near infrared, it could detect this near infrared fluorophore. So this is where we got really excited, and we backed up and said, okay, let's look at all the components that make up a fluorescence guided surgery system, and let's try to replace component by component with low cost, easy to source parts. So, to really spell out how this works. If you have a near infrared fluorophore, and that near infrared fluorophore is embedded within a tumor, if you shine 780 nanometer light, approximately, the fluorophores are all a little bit different in terms of their excitation profile, but 780 typically works, then you will have emission of that near infrared fluorophore at some higher wavelength, around 815, let's say. You'll also have some visible light, of course, that's coming off the fluorophore because your room lights are on. And you'll also have reflection of whatever light you have used to excite it. And so you need to do something to make the 815, that fluorescent signal, the signal that's important to you. You need to do something to isolate it. And so what you do is you filter out both the excitation peak, part of the excitation peak. You also want to split the visible and the emission peak together. And so there's some filtering that occurs. And now you've got two different kind of light sources available. One is the visible, one is the near infrared. And then you might want to have one camera that detects the visible and one camera that detects the near infrared. You want to combine those two cameras together. You want to display the resulting signal. And so we tried to see if low-cost laser diodes could be used as our excitation, if we could find low-cost optical filters to do the filtering step, if the Raspberry Pi cameras could be used. And there's one that comes in the one that's called the no IR. It has no infrared filter inside of it, so it can detect the near infrared light. The standard one has an infrared filter in it, so it cannot detect the infrared light. We found that the Raspberry Pi compute module was a really attractive option because you can actually feed two camera streams directly to it. And then for display, we saw if we could take an off-the-shelf headset, but repurpose it in a way that makes it much more powerful and would allow for this fluorescence-guided surgery stuff to be done. And we want the whole thing to be wearable and wireless. And in order to do that, we wanted to make sure that we can power it with a battery rather than having you connect it to the wall. So I'll jump through some of this stuff fairly quickly, and we can always go back if there are questions. We characterized these low-cost laser diodes, 120 milliwatt powered by 3 volts, and it costs $18. And if you put them into an array, you can get a fairly homogeneous, it's not perfect, but a fairly homogeneous 10 centimeter diameter area of excitation of 10 to 20 milliwatts per centimeter squared, which are safe values to use and the kinds of values that the more professional devices use in order to excite fluorophores commercially. And this is one implementation of it, kind of a rendering of it and the actual build of it, 3D printed components to hold the laser diode housings, something to hold cleanup filters, something to hold a white light source that can also be battery powered, and then a filter for the white light source as well. So a lot of interesting, very simple things that you can do to combine these laser diodes together. And then the question is, what about the Raspberry Pi camera? There are multiple now. When I did this work, there were only a few. Now there's even more, and it's really exciting. But this is just one to show you. One of the cool things about this Raspberry Pi V2 module is that there is a company that makes an extension cord that connects the little camera sensor, which is just 6 millimeters by 6 millimeters, tiny, tiny little thing, to the 25 millimeter square PCB board. So in some sense, you can really miniaturize the imaging module part by using these extension cords. And the power of this sensor, I mean, it's really quite amazing. Huge number of pixels, the field of view is fairly large. And we can do some experiments and to validate what is the typical resolution that we can achieve at a 50 centimeter distance, which is basically the distance from your head to the surgical field, and the camera is going to be mounted on your head, right? So we found that at 50 centimeters, we can get a 200 micron resolution, really exciting. And what's also really exciting is that the camera, the lens that's built is designed to be in focus from some distance to infinity, typically 100 to infinity. But if you kind of twist a little bit, you can find a region where you can get it in focus from 30 to, let's say, 60 centimeters about. And that's a great working distance to work with when you're a surgeon. You're not going to be holding it out. I mean, unless you're one of these really, really tall people, you're not going to be holding out a meter, right? You're going to be holding, the tumor is in your field of view, and it's going to be between 30 and 60 centimeters. So here's how we do alignment of the two cameras. It's really quite simple, honestly. You have a mirror, this cold infrared mirror, which you can get for $40. And it basically will take the infrared light and it will allow the infrared light to pass through, and it will take visible light and it will bounce it off at 45 degrees. So if you place this at 45 degrees and you have these two, the no IR camera, which has no built-in filter, and the visible camera, which has a built-in filter, place them at 45 degrees to each other, 90 degrees to each other. You've got light that comes in and then it goes right into the visible camera. And then the light will bend a little bit, and then the infrared light, and will go through and hit the infrared sensor. And if you put a long pass filter in front, then you'll filter out all of the near infrared light that's not emission, that's the excitation near infrared light. That's the basic setup. And this is a 3D printed model that shows you how you can construct it. So it's basically a a tiny 3D printed component that these sensors snap into. There, you slide the cold mirror and the long pass filter into it, and then you just fasten it with little tiny 1.6 screws, 1.6 millimeter screws, or M16 screws. It just stays in place and it fits, and it's great. It's tiny, it's like 1 cubic centimeter. So when you do that, this is a cutaway of it. You know, you've slid in the filter, you've slid in this filter, you've aligned it. This is kind of how it puts together. When you do that and you turn it on, even though you've done this careful work of making sure that you've taken account for Snell's law and you've taken account for exactly the position of the various sensors relative to each other, you take a picture, and this is a picture of a resolution target. And in pink, you can see the image from the infrared camera, and in green, you can see the picture from the visible camera, and they're kind of overlaying here. It doesn't align. It's like, why? You know, you look through, let's say, line pair 1, and you see you have 1, 2, 3, 4, 5 lines. Yep, I see 5 lines, but they're not aligned between the infrared and the visible. And the reason is, you know, things aren't perfect when you 3D print. I mean, the tolerance is not 100%. And the way that these sensors are made, maybe there's a little bit of offset between them. So this is an issue where, how do you take something that's not built to be perfect, but turn it into something that is perfect? And so the answer that we came upon was that we could create a transform between the images at, let's say, 50 centimeters to make them perfectly aligned, just a software-based transform, taking fiducial markers. And once we did that at one distance and apply that transform, it works at every single distance. So essentially, you do a one-time calibration that fixes this misalignment that's due to imperfections in manufacturing of the sensor or imperfections in manufacturing 3D printing. And now you fixed it and it works all the time, because the offset was purely an offset due to malpositioning. One calibration step fixes it completely. Then the next question is, okay, so what filter do you use, right? What filter do you use to filter out the excitation light while keeping the emission light? So here's a plot showing you the intensity of ICG emission, ICG being the fluorophore that we're interested in. And many fluorophores have in the near infrared have very similar peaks around, you know, you have a peak from 810 to 820, somewhere around there. And the excitation usually is around 780, though you can also excite at 770 or 760. Basically, the closer you get to 780, the better signal you get, but you'll still get some fluorescence even if you excite at 770. So ideally what you want is an optical filter, and here you see the optical densities in filters, that has really high optical density below where the ICG peaks. So you can see this Semrock 370, which costs $375, has high optical density up until 810, and it starts dropping down. So it lets light in that's like 820 and higher, and blocks out light that's 810 and lower. And so if these are various combinations of filters, we tried out a lot of different ones. We found that if you take this Newport long pass filter, which costs $38 individually, one of them has fairly low optical density. It doesn't work that great. But if you double them up, just put them right next to each other, as you see here, you get performance that's on par with a much more expensive filter. So this was another trick that we looked into to try to make this system more affordable. And this is a characterization of the fluorescence sensitivity that we have using these various different filter combinations. And this is just using an assay with the IR 125 solid phantom of varying different concentrations. We were really amazed to see that we can detect, this is signal to background. So if the signal to background is 1, then the signal is the same as the background. You really can't detect anything. We can detect a signal to background of 10 for some of these filters for concentrations of 5 nanomolar, which is really quite low. And that's what the really industry-leading devices are able to capture. We could even get down to 1 nanomolar for the double 830, which is the lowest cost option other than the single 830. The single 830 didn't do so great. I mean, for higher concentration, it could still work. But if you're going down to as low as 1 nanomolar, the double 830, which is only $76, worked similarly to the much more expensive options. And so then we did an animal study to just validate that our system can detect what we think it can detect. And this was a tumor targeting agent that Dr. Sam Chilafu developed called LS301. This is a compound that's currently under clinical trial at UT Southwestern, a phase 1 trial. It started here at WashU, moved over to UT Southwestern when he moved over there. And so we're testing it in humans with breast cancer. But this is a test in a mouse model of breast cancer where we create a tumor, the tumor grows, and then we inject this compound and we check 1 day later to see if we can see accumulation of the compound. And sure enough, we can see accumulation of the compound right into the tumor. And depending on which filter you use, you get a better answer. But just to point out here, using that double long pass filter, $76 of optics, we get a signal to background exceeding, between 6 and 7 in this area of tumor. I mean, that's a really, really strong signal. And we only did 4 animals because the signal was so strong. It was like, well, we don't really need to do any more. I mean, there's a clear difference here. You can obviously see that the tumor is there. So a little bit about, we can talk about how do you actually connect all the pieces. You can connect the cameras as USB cameras. You can connect the cameras as along the CSI port and see a direct stream right to the device. You can have electronics that are controlled by the pins of the Raspberry Pi. And this was just a simple board that we soldered together that consists of switches that can be used to control the lasers to turn on and off, or to oscillate and to go on and off. And I'll show you that in a second. Things to control a cooling fan to keep the device operating at normal temperatures, at safe temperatures. A switch to turn a white light on so you can actually see where the area of interest being visualized with the lasers. And one point to make is that when you when you just turn the laser on, you'll see that there's if you don't have a good filter, you'll just see the laser itself as a fluorescent signal. So this is kind of back to why it is that we want to have good filters. We want to have good filters because you want to avoid seeing this circle. But the other thing I want to point out is when the laser's off, see, we have this like cardboard box and a piece of white paper above it. The laser's off and you're looking with room lights on, you still see a signal. You see this white paper, you see a little bit of a glow. And that's because there is background near infrared. If your windows are open, which they might be in a setting of somewhere where you're doing surgery, or if your room lights have a little bit of an NIR tail, you'll get some near infrared being picked up, even with the filters that you're using. And so one thing we try to see is, could we potentially have a means of removing the background signal? And this is something that we found could be done if you pulse the near infrared laser, the excitation laser, if you pulse it so that it turns on at the end of a frame being read in. And so by doing that, you could have one frame off, one frame on, one frame off, one frame on. And if you timed it incorrectly, if you left the laser on too long, then every frame looks on. And that's just a consequence of the electronics, and I can explain that. But the point is, you can find the right timing so that every single opposite frame either has background included and no excitation, or has background included and excitation. And then if you take one frame minus the other in sequence, then in real time, you can have background subtraction. Again, this is something that the sophisticated devices can do, but it's, to me, quite remarkable that something as simple as the Raspberry Pi can achieve this in real time. And then I'll leave you, I want to cover this point and obviously leave time for questions. But one of the most important things to think about if you're making a heads-up display that's detecting something and also displaying something is that from the perspective of a surgeon working at fairly close distance, if you are not aligning the image that you're showing to the surgeon with the real world, then disaster can strike, right? The surgeon can just cut the wrong part of the tissue because what they're seeing, maybe it's aligned within the cameras, but it's not aligned with their eye. And so one idea, that we started with initially was, well, let's just put the camera in the middle of the surgeon's eye, like in the forehead, and it'll just look at the scene, and we'll do a software correction, right? Like the eye sees this, we put a camera behind the eye to see what the eye sees. This is what the eye sees of the target. Here's what the camera sees of the target. They're not the same, but we can create an alignment to make them the same. The problem is that if the alignment is done at 50 centimeters, but then you start looking 30 centimeters away, the alignment is broken. And you can see that the alignment only works when you're at the distance where you set the alignment. And then when you start moving away from the alignment, right, we're aligned, now we're not aligned. And if you characterize the error there, you'll see that the distance, the amount of error in the versus horizontal or vertical in this case is quite high, 5 millimeter error, 15 millimeter error once you're 10 centimeters away from the alignment distance. That's not going to work. That's going to be really dangerous for a surgeon. And you could say, well, okay, fine, let's do better. Let's just put it right between the eyes. Well, that's good. You're aligned at 50 centimeters away. But again, as soon as you start moving away, you get a misalignment between left and right. And you can quantify that. So now we fixed the vertical error problem, but the horizontal error problem is still there. So we came up with a different way of doing it. And as far as I know, in terms of doing dual visible and near infrared imaging and co-alignment with the eye, we think that this is the first implementation of this. What if you may put a beam splitter in front of the eye so that the light that's coming off of the target either goes directly into our dual camera system or passes to your eye? In this case, there is still some alignment that has to be done to fix, vagaries of the actual mechanical alignment process where, like, is the camera really centered with your eye? But now when you take the video and you move away or towards the alignment distance, everything stays aligned. And so in this case, the error that we find is really negligible. And so this is an exciting thing that, that we did to try to really ensure the most important thing when it comes to fluorescence-guided surgery, if you're using a heads-up display, is that you have perfect alignment. This, I think, is a really neat way of doing it. I think it could be incorporated in a lot of different technologies out there that people are working on for heads-up displays. So the RoKit Air is a really cool media-based virtual reality or augmented reality tool. You can buy it on Amazon for $400. It's got birdbath optics, which is like, the light comes in from above and then bounces off. But what's neat about it is, first of all, the specs are really cool. 43-degree diagonal field of view. That's kind of similar to the HoloLens, but it's only $400. High resolution for both screens, very light, 83 grams. Also, it's kind of hackable. Like, you can take it apart. You can disconnect one of the screens if you just want monocular vision instead of binocular vision. We 3D printed a component that directly mounts to it, and then we can mount stuff on top of it like we do here. So then we can easily mount our beam splitter idea. We can mount the laser on the forehead. And if you're looking through it, I'll show you later what it looks like. But this is kind of what it looks like mounted on someone's, on a mannequin. So this is what it looks like when you look through the RoKit. I mean, you can see the Raspberry Pi operating system just right there. Sorry for the shaky camera. And if you want to see what it looks like when you're wearing it, this is what you see on the Raspberry Pi. One of the neat things, for people who haven't worked with heads-up displays, if you on your head-up display show something white, you really can't see through it. But if you show something black, you can see right through it. And so as long as your interface basically has a black background, you can then superimpose whatever you want on top. You can see here, I'm looking through it. I see my hand moving, but then I see the superimposed display from the application. And so here, what you'll notice is we will start the application running, and it'll open up a window. And then that window, when it loads, will be black. And now you've got an interface, but you also see something superimposed. We can also talk a little bit about, and this is just open discussion, how do we achieve calibration between the surgeon's eye? How do we ensure that the surgeon and the camera are aligned? We do that through a series of steps with a fiducial marker. And I can show you this if you're interested. And this is just showing a little bit of what it looks like when you actually turn the laser on. Here's a vial of ICG, and it's there. This is the visible stream. This is the near infrared stream seen by a remote observer, which our software can allow. But also, this is what the person sees looking through the goggles, and you see the thing move around and the infrared tracks it. So this is the first step towards a bigger project, which is really how do we make fluorescence-guided surgery available to an audience beyond the academic centers like WashU and others, where we have tons of resources and we can buy hundreds of thousands of dollars of equipment and also service contracts. How can we take technology that's available off the shelf and turn it into something that's really quite powerful? And maybe part of that is these kinds of open source ideas. At least this work shows that seemingly inexpensive and low power technology is actually quite powerful. You can do quite a lot if you put it together correctly. And I've built videos to show like how one could actually construct this piece by piece. Maybe something like this could be useful one day to help people build it on their own. But I think there have to be a discussion and safeguards in place and all sorts of things that I'd love to talk to people who are interested in this work on how do we allow this kind of technology to be accessible to the world while also being safe and ensuring that that we're not causing more harm by creating something that really gives people a false sense of security or a false sense of where the tumor is, and they cut something wrong and then harm is done. So with that, I'll take any questions. Happy to engage in the conversation, and thank you again for the opportunity to speak.
Thank you. It was a fantastic presentation. We already have a couple of questions coming in on the Zoom, so I'm going to start with those. We have a question: can the system you've created potentially provide stereoscopic images for better depth perception? And given the way that Stephen introduced me, I thought I would interject there and say, like, is there a clinical benefit to depth? Of course, being a XR person, I went right to that. I've tried a number of monoscopic kind of monocle displays over the years, and I'm pretty familiar with them, and I could see how even in mono, this would be a great project. But is there clinical benefit to like a stereo view here? And is there a performance challenge that would be presented by the Raspberry Pi platform?
Yes, great question. Great question. So, okay, so ideally, this is this is stereoscopic, and we can make it. One could make it stereoscopic, and there are plans to make it stereoscopic by essentially, you can imagine having another beam splitter over here and a second camera, right? And that is possible, and that is doable with the Raspberry Pi system. With the Raspberry Pi, I'll kind of give you a sense of what people have done with Raspberry Pi. There are people who've done quadrascopic cameras with Raspberry Pi. One way that you can do that is a secondary board that, and this is available from a company called Arducam. They take the stream from all 4 cameras and they downsample it so that the final stream has the same size as one full resolution image, but each quarter has one view from each camera. And so there is a trade-off. There's always trade-offs, right? But there is a trade-off there that in some cases may be quite reasonable because it gives you that stereoscopic view. Another aspect to consider is the way in which we've built this. We have both visible and near-infrared imaging being done simultaneously. And really, if you're a surgeon, the visible is what you see, right? You already see the visible. So you don't necessarily need to, and in fact, it would be very weird to superimpose visible on top of visible. You'd only want to see the near infrared superimposed. The reason to capture the visible is that if there's a remote viewer or you want to record the session, you want to have context for that recording. So you need to have the visible there. But in some sense, what that means is you only need one visible camera because the visible doesn't have to be stereoscopic, but the near infrared you need 2 because the near infrared to be superimposed needs to be stereoscopic. So that's one point. The other point to say is a lot of this comes down to the different devices that you have. I kind of glossed over this point, but as you can see here, this device, this Rokid Air, it actually has 2 independent screens here, right? This is one display, this is another display. When you just turn it on and you plug your computer in and you use it as an HDMI monitor effectively, it is essentially a very wide monitor. Like if you bought yourself a 3840 by 1080 monitor, that's what this device is, right? If you were to feed in data that happens to be encoded for 3D, then the software on this device actually displays it. So if you watched a 3D movie on this, your left eye and your right eye would show different things. When you just plug in the Raspberry Pi to the USB-C port of this device, it shows left and right mirror images. That is a disaster if you're trying to use it as a surgeon, because your left eye and right eye would see the same overlay, but in reality, your left eye and right eye should see different overlays. So that is why one implementation of this, I literally disconnected one of the displays to turn this binocular device into a monocular device. And in that case, as I experienced it myself, it was not a problem, because while only one eye has the near infrared overlay, your brain figures it out and you know where you're going. And oftentimes in surgery, you have a surgical plane you're working with, and so the depth isn't as much of an issue. However, I have found that if you are clever in terms of the settings on your Raspberry Pi and the kind of HDMI stream that you send to this device, you actually can program it to send as if it's one very wide screen. And then if you were to send the left camera to the left of the screen and the right camera to the right of the screen, you actually can generate 3D imaging and stereoscopic imaging. So it is possible. It does require interfacing with the device in the right kind of way. I don't know if all devices allow that. It's all off-the-shelf devices, rather. I did recently see a preprint from someone at Cornell, first author Matt Isaacson. They built a VR device for mice to track so that mice could look into the VR. And they used a Raspberry Pi, and they used two separate SPI screens and were able to send one video stream to the left screen and one video stream to the right screen. So I think it's possible to either have one screen that you split in half and send different streams to left and right, and then port that into a device like this, or a screen of your own making. You could create birdbath optics yourself. They're not that hard to make. Or you have two separate screens. And in that case, yes, Raspberry Pi allows you to do two double HDMI output, but I think performance-wise, you'll be better doing it across SPI and using like the TFT type screens that you can have. So there's a lot of options here from an engineering perspective of what one can do.
Yeah, fantastic. We have another question, looks pretty clinical in nature, so I wanted to go there. The person's asking, the use of tumor-specific chromophores is really cool. How much deep does near IR penetrate normal tissue? We need to excise tumors with a macroscopic margin of normal tissue. Yeah, absolutely. So
so estimates vary in terms of how much NIR penetrates. People say between 1 and 2 centimeters. I think that's not unreasonable. Some of the things that will change that number is the amount of scattering that you will have. And that will depend on the type of tissue that you're imaging. I will say one of the things that is kind of neat about doing this real time is that, let's say the tumor, is 4 centimeters deep. Well, the surface, as you open up the surgical cavity, you'll see a tumor. There will be a bulk tumor there. You start cutting, and let's say you've cleared, 2 centimeters of tumor. Well, now what's left in the cavity, again, you get 2 centimeters deeper than that. And so you're kind of iteratively going. So initially, you may not see the whole tumor because NIR light will only penetrate so deep. But as you clear out cavity, you will see deeper and deeper. Now, if there's a met that's beyond the, if it's patchy and you have tumor here and then no tumor and then 3 centimeters deeper there is tumor, that will be difficult to image with near infrared. I will say one of my interests my primary interest in my lab is the shortwave infrared, which is an even longer wavelength than the near infrared. In the literature, it's a bit confusing. Some people call it NIR2, but it's basically 900 to 1700 nanometers. People are working on fluorophores in that range. Once we have those fluorophores, we will be able to achieve even deeper imaging because there's much less scattering in the shortwave infrared. Another thing I would want to mention as someone who studies a lot of skin cancer and imaging with skin, this kind of imaging is not immune to the impact of pigmentation. Melanin will absorb near infrared light around 780 nanometers, like the light we're using here, and it will absorb the light that's coming out, the 815 fluorescent light that's emanating away from the tissue. So there is an important role a discussion to be had about ensuring that when you test these devices, you test them in a setting where pigmentation can vary from light to dark pigmentation. That means, being careful about measuring what the pigmentation is in the specimens you're using, or incorporating pigmentation within those optical mimicking phantoms the skin mimicking phantoms that you're using. Of course, with surgery, once you cut away the skin, that's not an issue anymore. But if you're thinking about doing this with skin intact, considering the impact of pigmentation, especially if you're thinking about, ensuring not just equity here in this country, but in the world, that is a critical component to consider.
All right, I'm going to stick with the imaging related questions. Next one is, what's the laser beam size on the patient? I'm trying to understand if you have to scan your head to excite the IR dyes in the ROI.
Yes, it's about 10 centimeter diameter. And so as you move around, it'll just be in your field of view. I will say that is, I will fully admit that is a weakness of the current implementation, and expanding the size of the ROI would be very welcome and great. I think, another alternative method would be that you set up an illumination field separately, like on a tripod next to the tumor area that you're working on. That would allow you to, have a larger field of view, use a larger power laser. There's other things that one could do. There's people who've tried to do this with halogen lamps because they're quite bright and can have a large field of view. Really have to filter those very aggressively because they're very, very broad spectrum. I've looked at also trying to use really high power LEDs. So I do think that there's room to grow. This particular application was purely like, you have a surgical field, it's a fairly small area, and you're working one step at a time. And so where your hands are, that's where we want the laser to be, laser illumination area to be. And 10 centimeters diameter seemed like a reasonable starting point. But yeah, if we could expand it, that'd be great. It does require, if the tumor is larger than that, for you to move your head around. At the same time, your head and your hands are moving around as a surgeon. And so we thought that wasn't a terrible trade-off to take. So that was the design of the system. All
right. Next question I thought was interesting. Have you considered adding some simple voice commands for control? Certainly there's good precedent here in surgical robotics and in other applications. A lot of the uses of the HoloLens are taking advantage of the great text-to-speech capabilities there. Is there room for that kind of interaction, particularly on the Raspberry platform?
Yeah, great question. Great question. Voice control on the Raspberry Pi is something I have not played with. I think it is possible. I will just step back and say the software that I showed is my humble attempt as not a professional software engineer to show some feasibility. So the way we did this was the Raspberry Pi acts as a server, and this is a Flask app. So it's a Python server, and then the Raspberry Pi literally in a browser window opens up an address, and that address is the application that allows you to do calibration, turning lights on, turning lights off, and within that window, that's where you see all the superposition. The server also provides an address called remote, which allows a remote viewer to see a totally different view, right? So the surgeon sees one view, the remote viewer sees another. There's precedent for doing voice command, yes. I would have to look more into whether the Raspberry Pi can manage it. There are, in the implementation that we have at UT Southwestern, there is a foot pedal that essentially, if you can imagine like a trackball mouse with buttons, it's kind of like that. So it does allow the surgeon to, without using their hands, make inputs and outputs on the device. UI is, it's a big field, and I think, there are lots of opportunities for making all sorts of interesting UI enhancements to this. I'll give you an idea that one person shared with me when I presented this at a conference. He said, with the laser, in order to improve safety, you could have it so that the visible camera, when it's looking at the surgical scene, if it sees some sort of fiducial marker, then it knows, oh, I'm looking at the surgical scene, and therefore turn the laser on. And if I don't see the scene, if I don't see that fiducial, laser cannot turn on, right? So if the surgeon looks up, the fiducial is no longer there, and now the laser turns off, right? So like these are there's a lot of like clever things like that one can do when you have that extra video stream. And so whether it's using some sort of hand gestures or whether it's audio, I think there's a lot of opportunities for UI.
Absolutely. Next, we have a question about FDA certification. And I'd like to expand a bit upon that and just see if you've, along the way, done any documentation, such as a design history file or a software development plan that might assist with that process. And also just get your comments in general on how you feel the FDA perceives the maker movement and some of the topics you discussed.
Yeah, it's a great question. I'm not an expert in device approval, right? I'm a kind of a hacker or a maker, and I know that there's a lot for me to learn. So I'm happy for people to give me this pushback. One of the things I will say is that just showing that low-cost devices can give you such strong signals is, I don't know, it's invigorating to me, because it tells me that there is an in-between between super expensive devices that effectively no one in the world has access to and nothing, right? There is some sort of in-between, and we have to work together to find a way to get to that in-between in a safe way. I think that when it comes to using devices like this for an FDA-approved means, sure, we have documentation of how we do this and all the papers and all the stuff, but will they trust that an off-the-shelf thing like Raspberry Pi has the has the specs necessary to like not turn off all of a sudden, or there's all these things that we build into really FDA-approved devices that may not fly when it comes to using a Raspberry Pi. So I don't know that I'm confident that it definitely would work. I do think Raspberry Pi, the CM4 maybe has those specs. I honestly would need to look more into it. But when you combine, the Raspberry Pi is one thing. Taking a off-the-shelf laser diode, that is low cost and cheap and has some sort of, probably that's not going to fly, So I think I look at this as inspiration for a version that could be FDA approved. At the same time, I don't know. I don't know if people on the call will share this feeling, but a lot of my thinking around this changed with the pandemic, because in a crisis situation around the world, people were, 3D printing face shields and creating all sorts of, innovative ways of saving oxygen and doing all sorts of things off the fly. Like, if you're a fan of MacGyver, kind of like MacGyver, the world was full of MacGyvers, right? Like trying to do what's right in a safe way. And it just hit me that we need to have better examples out there of people just saying, well, what can I make with this simple stuff? What is possible? It's not, we're not in a crisis all the time. And so it's not reasonable to take a risk on a patient with something like this, but maybe this inspires the work that's necessary to get to something that is viable while also not being super expensive. So, if I was to say, how would I take this into a global context? I would have to want to partner with someone who actually works with a community, like a local manufacturing partner, and they have expertise in terms of what is it that needs to be done in order to ensure safety and efficacy with their local hospitals. And we would work together, and I'm sure I would learn a lot in that process in terms of, well, maybe we don't source the components from laserlands.net like I did for this, but we find them from somewhere else. But the prices aren't going to suddenly skyrocket 100x, right? So that's where I think that I don't want to say that this is the answer. This is the path towards the answer in terms of showing what is possible with stuff that on the surface might look like is not powerful enough to achieve the goal. But yeah, the process of doing this for FDA certification, that would be the expensive part, right? To really get FDA approval, I mean, that you can't do that on the cheap as far as I know. But if there are people on the call who have ideas of how to make that happen or create a pipeline of tools and techniques that can be used and reused, I think that would be really powerful for the global health community, because I think, if that's the thing that stands in the way, then there will always be this super expensive burden that is going to be hard to cross.
Yeah, and the FDA has this breakthrough medical device designation that they can apply. And I think in this case, although this is technology that is, tried and true, doing it in a small form factor that could be put in a box and carried on a plane just about anywhere, doing it for under $2,000 are both, I think, pretty big breakthroughs. So I'd like to thank you very much for your time. We're just coming up on the end here. Really enjoyed the presentation and looking forward to following up on your work as it progresses. Thank you very much.
Sure thing. And I'd be happy to follow up, by email if there's questions that were unanswered. I'm happy to share my email and answer if there's other questions.
And thanks to everyone who joined today. And be sure to share the videos when they're available on YouTube and LinkedIn with your colleagues and anyone who might have missed the show. Thanks very much, everybody. Cheers.
Yeah, just to wrap up, the video will be up within a week, probably sooner. We usually beat that schedule. And we'll send out a note that it's now available. This was a fabulous talk. One person remarked that it reminded that Leo reminded him of Tom Furness from the University of Washington. But his maker ability reminded me of Lon Hacker, who went between multi-million-dollar research contracts for NASA to building low-cost data acquisition devices. But what he was really doing is forward researching devices that he would use 10 years later in his low-cost data acquisition devices. And the understanding of what's available for components and how they relate to the whole was fantastic. This was a great talk. There are people, Leo, who were interested in collaborating, and I will make sure I connect you with them, them later on.
Perfect. I did see I just wanted to say one thing. Someone in the chat asked about the chemical compound. So just so I'm fully, I don't want to confuse anyone about this. Some chemical compounds are already available, like ICG, indocyanine green, FDA approved. It's a non-specific marker of vasculature. It's used all the time. So that is one option, and that's what was used in the original studies. The compound that Dr. Chilafu developed, LS301, that is a new compound. It's being evaluated for safety and efficacy as we speak in an ongoing trial at UT Southwestern. We think it's really powerful. It targets phosphorylated annexin A2, which seems to be upregulated in the tumor microenvironment by activated fibroblasts. It is one of many compounds right now in the field that are being developed that are tumor selective. This fluorescence-guided surgery field, I think, will continue to have new compounds created and new devices created, because it just kind of makes sense that we should be able to see what we're working on. And that includes two compounds that target the nerves, so that if, let's say, there's a gunshot wound and you're doing surgery and you want to make sure that you clear it out, but you don't cut a nerve. If the nerve was glowing, man, that would make life so much easier. So there's a lot of these kind of ideas out there. It's an exciting time for this field.
Wonderful. Well, thank you for coming. Thank you, Jeff, for moderating. And Leo, thank you very much for presenting your excellent paper. We really appreciate it. Thank you.
Thank you again for the chance to talk.